Bismillahirrahmanirrahim. This is Dr. Eskam. Hope that you are doing well. So today we discover a new stage of embryonic development, which is fertilization stage. So let's know what is fertilization stage. The fertilization stage is the meeting between the male haploid gametes, which is the sperm, and the female haploid gametes, which is the oocyte. So the sperm is formed by spermatogenesis, and the oocyte was formed by oogenesis. So this meeting is done in order to form one single diploid cell called zygote or fertilized egg. Okay, so this is fertilization. According to the location of the fertilization, we have two types of fertilization. In fact, if the gametes meet inside the body, we call this kind of fertilization internal fertilization. So it concerns many examples, such as the mammals in general. So mammals in general used internal fertilization. But if the gametes will meet outside the body, it's called external fertilization, and it concerns several examples such as fish and urchin. So for us, since we have taken as example spermatogenesis in men and oogenesis in women, so let's start by the internal fertilization. So we will discover its definition. After that, we will talk about its location, and we will see the characteristics of the gametes before fertilization, and finally we will talk about the stages of fertilization. So let's start by the first point. What is internal fertilization? So the internal fertilization is the internal meeting between the sperm and the oocyte. Of course, in order to form the zygote, which is the diploid single cell. Okay, so when I say internal meeting or inside the body, I mean the body of the woman, of course. Okay, so in the body of the woman, exactly in her female reproductive system, the fertilization will take place in the fallopian tubes. The fallopian tubes are located near the ovaries. Okay, so the meeting between the oocyte and the sperm will happen in the fallopian tubes. If we observe the fallopian tubes, it happened in the beginning of the fallopian tubes. But you have to know that the spermatozoa aren't deposited directly in the fallopian tubes, but they are deposited in the vagina, and they go through the uterus, enter the fallopian tubes, where they will meet the oocyte. Okay, so since our two cells will meet, let's talk about their characteristics to make a good fertilization. So we will talk, of course, about the sperm, and after we will talk about the characteristics of the oocyte. So let's start by the sperm. Here you have, in this picture, you have the microscopic, the electronic microscopic picture of the human sperm, and here you have its diagram. So as you can see, the sperm must be composed by three important parts. Okay, so the first part is called the head. Okay, and there we must find two organs. The first organ is the pocket, one pocket called the acrosome and containing enzymes, and the second organ is nucleus. Okay, after that, this part is called intermediate piece, and it contains a lot of mitochondria. So as you know, mitochondria is used for production of energy to make the spermatozoa moving. Okay, and the third part is flagella, and the flagella is used for moving from the vagina until the fallopian tubes. Okay, so now let's talk about the oocyte. So the oocyte, as you know, is an oocyte 2 blocked in metaphase 2. After the ovulation, this cell will be ovulated with two layers. The first layer is pellucid zone, which is a layer of glycoproteins, and the second layer is corona radiata, which is a layer of follicular cells. Okay, so when the spermatozoa arrive around the sex cell, they must find her like this. Okay, so let's now start talking about the stages and discover how the sperm will fertilize the egg. So firstly, the sperm have to cross the first obstacle, which is corona radiata, and then it have to cross the second obstacle, which is pellucid zone. After that, it will cross the third obstacle, which is ovular membrane. And then two new events will happen. The first event is blocking polyspermy, and the second event is fusion of nuclei. So let's follow all these stages, and we will start by the first one, which is passage of corona radiata. So, as you know, the corona radiata is composed from follicular cells, and these follicular cells are disposed all around the sex cell. Okay, that's why when the spermatozoa arrive in the area, they can't cross this layer of cells. But something important will happen. In fact, the concentration of the spermatozoa in the environment, when it is important, it provokes the detachment of the follicular cells. Okay, so the spermatozoa concentration must be important to provoke this detachment, in order to create some passages for spermatozoa and allow it to go in the contact, to arrive in the contact of the pellucid zone. Okay, so if the concentration of spermatozoa in the area is less than 20 million of spermatozoa, the follicular cells don't detach, and the spermatozoa can't cross these cells. Okay, so that is how the spermatozoa will cross the corona radiata. Okay, but after the corona radiata, they arrive in the contact of pellucid zone, and here it is the second obstacle, and the spermatozoa can't cross this layer of glycoproteins. Okay, so each sperm must find a way to cross this layer. In fact, each sperm has in his head a pocket called acrosome and containing enzymes, and of course enzymes will destroy the pellucid zone, but the problem is that acrosome is closed. Okay, so that's why the spermatozoa must be fixed before, and then they can't they can cross the pellucid zone using their enzymes. Okay, so let's see this picture. So here we have taken only one sperm. Okay, so when the sperm arrive in the contact of a pellucid zone, it has to be fixed. Okay, in fact it will be fixed in specific zones called ZP3. These zones, these fixation zones, are situated in the pellucid zone. So each sperm must search for these fixation zones, and when it finds one fixation zone, it will be fixed using a special receptors called RZP3. These receptors are located in the membrane of acrosome. Okay, so when I say ZP3, it's located on the pellucid zone of the oocyte, and when I say RZP3, it's located in the membrane of the acrosome. Okay, so this is the first process, which is the fixation of sperm. This process, this fixation, provoke the opening of the acrosome, and of course the acrosome contains enzymes, so the opening of the acrosome provoke the freedom of the enzymes to digest a part of pellucid zone. And this process, so the opening and the digestion of pellucid zone, is called acrosome reaction. Okay, acrosome reaction, and by the acrosome reaction the sperm arrive in the contact of ovular membrane. Okay, so what's happened after? So when the sperm will arrive in the ovular membrane, it must find another way to cross this ovular membrane. In fact, the sperm will use another type of receptors called fertilization. Okay, so this fertilization will be linked with another type of glycoproteins called integrin. So the integrin are glycoproteins located in the ovular membrane. Okay, and fertilization are located in the membrane of the sperm. Okay, so this link between the fertilization and the integrin provoke the penetration of the sperm in the oocyte cytoplasm. Okay, you have to know that it's only the head that penetrates in the cytoplasm of oocyte. The other organoids, I mean intermediate piece and flagella, will degenerate. Okay, so this process is called adherence of the gametes. Okay, so when the head of the sperm penetrates in the cytoplasm of oocyte. Okay, so what's happened after? Don't forget that in the area there is not only one sperm, so all the spermatozoa are in competition in order to fertilize this egg, but the egg has to be fertilized by only one sperm, and it will be the fastest. Okay, so that's why after the penetration of one sperm, all the other spermatozoa have to be blocked by the oocyte. Okay, that's a new event called blocking polyspermy. So polyspermy is to be fertilized by several spermatozoa. okay, and it can't happen. So that's why the oocyte must block all the other spermatozoa. Okay, in fact, the oocyte will proceed by two kinds of blocking. So after the penetration of the fastest sperm, the oocyte will use what we call rapid blocking. Okay, this rapid blocking happened very quickly, and it consists on a perturbation of its own concentration of some ions like sodium. Okay, and it happened very quickly. That's why we call it rapid blocking. Okay, and we can also call it ionic blocking because we use the perturbation of concentration of some ions. Okay, but this blockage is not. That's why the oocyte will proceed by another kind of blocking, which is called slow blocking, and consists to the formation of this envelope called fertilization envelope or fertilization membrane, and which don't let any other sperm penetrate in the oocyte. Okay, in the oocyte cytoplasm. Okay, so let's see how it happened. So in fact, when with the penetration of the first sperm in the oocyte cytoplasm, look at the membrane. Look at here to the membrane of the oocyte and the plasma membrane of the oocyte. In fact, the oocyte has two membranes. The first membrane is the ovular membrane, and the second membrane is the plasma membrane. Okay, so under the plasma membrane there is some structures called cortical granules. Okay, so after the penetration of the spermatozoa, the cortical granules will move from this place, which is under the plasma membrane, to this place, which is the space between the plasma membrane and the ovular membrane. Okay, so when the granules move to this space, they transform the membrane, the ovular membrane, to fertilization membrane or fertilization envelope. Okay, so it starts from the point of penetration of spermatozoa and continue all around the oocyte. Okay, so it happened very quickly. That's why after only 1 minute, all the fertilization envelope is already formed. Okay, so it starts from the point of the penetration of the spermatozoa and continue all around the oocyte. Okay, and this envelope can't allow to any other sperm to penetrate in the sex cell. Okay, so this blockage is called, as I said before, slow blockage or mechanic blockage, because with the formation with this envelope, it's a mechanic obstacle for other spermatozoa. Okay, so after the formation of this envelope, the oocyte 2, which was blocked in metaphase 2, finishes its meiosis and transforms to egg cell. Okay, with this nucleus containing 23 chromosomes. Okay, indeed the spermatozoa will penetrate with its 23 chromosomes. Okay, and but the two nuclei will come together and fission in order to form one diploid nucleus, which is the nucleus of the zygote. Okay, and of course the zygote will start directly a new stage of development, which is segmentation stage. Okay, so hope that you understand me, and I'm waiting for all your commentaries or all your questions. Don't forget to contact me on Telegram and on Facebook. See you for the next video.